My honor to introduce today's speaker, Professor Peter Holland from Oxford. And Professor Holland obtained his bachelor degree in zoology from the University of Oxford in 1984. He then went to the National Institute for Medical Research at Mill Hill, London, and obtained a PhD degree at the University of London. He then held research and teaching positions at the University of Oxford and was one of the founders of the field of the evolutionary developmental biology. In 1994, he was appointed Professor of Zoology at the University of Reading and became the youngest full professor in the UK. And in 2002, he returned to the University of Oxford as the Lineker Professor of Zoology and served as the department head from 2011 to 2016. His research addressed the fundamental questions of how the evolutions of animal diversity can be explained through the evolutions of the genome. In particular, he's very interested in the evolutions of Homeobox genes and their functions in different animals. Throughout the years, Professor Holland has received many awards for his great research contributions. And in 2003, he was elected as a Fellow of the Royal Society. And today he's going to give his third lecture of the Jung lecture series. And without further ado, let's give a warm welcome to Professor Holland.
Okay, can everyone hear me? Is this microphone on? Yes? Excellent. Those of you who need a seat, there's going to be, it should be okay. Yeah, 2 more seats at the front. What I want to do today is think about this question of similarity and difference between species.
We have stairs, I know.
Yeah, okay, good.
We have stairs.
So let's think about where the structure of the animal comes from. It's always been thought of, but let's think about what the really difficult questions are in biology. Things like the functioning of the brain, clearly it's a difficult question. But this is also a particularly interesting question, which is how the 3-dimensional form, the complexity of an animal, is a single cell. Pretty much every animal species will start life as a single cell. So we'll have some examples here. And from that single cell, which divides into 2 cells, 4 cells, many cells, of course those descendant cells need to differentiate into particular types, tissue types, and get organized in a particular way. So it's a very complex process to try and think about. That enormous progress has been made in the last 100 years into these problems, how a single cell becomes a multicellular animal. And really that progress has been made primarily by people studying these sorts of species. These I would call the main model species in animal developmental biology. We all know what they are, and we know what sorts of experiments have been done. Pull apart interesting aspects of developmental biology from these species. But what those analyses in flies or worms or mice struggled with really was similarities between those. I put those question marks there to indicate that over a very long period of time, research was primarily finding out about developmental processes in mice, or in flies, or in nematodes. And for a long time, there were questions about how can we actually compare between these species? What do we do if we want to make comparisons? And to my mind, the biggest breakthrough in this field was the discovery of the group of genes called Hox genes. Now, I'm sure that almost everybody in the room knows what Hox genes are, but I just want to emphasize how important the discovery of Hox genes was for thinking about developmental processes in animals in an evolutionary context or a comparative context. So what we're dealing with here, what you can see at the bottom, are 2 pieces of mutant flies, where what's got wrong with these flies, the one on the left-hand side, of course, the antennae, the cells which should have formed the antennae didn't form antennae, they formed legs. They got their instructions wrong. They thought they were in a different part of the embryo. The one on the right-hand side
a segment which doesn't normally produce wings has produced wings. They got their instructions wrong. They think they're in a different part of the embryo. And Ed Lewis, on the top left-hand slide of this picture, was awarded the Nobel Prize for his work on those sorts of genes. That's flies, of course, and I think, as we all know, the genes which encode, which have been mutated in those flies, are the homeotic genes, also now known much more commonly as the Hox genes, which tell cells where they are along the body axis. And of course, what the work of many people, some of whom are shown on this slide, if I can get the pointer to work, shown at the top up there, are a range of people, some of whom, all of whom and many others, contributed to the discovery that this set of genes are found almost throughout the animal kingdom. Okay? So it was this line of work that I was involved in as well, and that what these genes are doing as we go across different species is telling cells where they are along the body axis. So that if we look across the animal kingdom, there is this common set of genes involved in the development, involved in embryonic development, with a particular role. So that's a deep, deep similarity between species. It doesn't stop there. Over time, other people, many researchers discovered other deep, deep similarities in development. Here's one, which is conservation of genes for telling cells the dorsoventral axis. There are also common genes for specification of eyes, on the top left-hand side there. Genes which seem to be involved in the development of hearts, or at least pulsatile contractile vesicles, both in flies and in mice, the same genes. Work in my lab with collaborators discovered a set of genes which seem to be involved in specification of regions of the gut across many different sorts of animals. So those sorts of discoveries highlight deep similarities in how embryos form in different species. That's all very well known, but what I really want to do in this seminar is talk about differences. Because although we've got deep similarities in the embryos of all these different species, it's still the fact that a mouse egg develops into a mouse, a human egg develops into a human, and a fly egg develops into a fly. A fly egg does not develop into a human. So these similarities, on top of these similarities, must be many, many differences in development. Evolution must have modified processes such that development leads to different outcomes in different species. So what do we mean by these differences? I like to think of it like this flow chart here. On the left-hand side, what we've got is the DNA. In the DNA are called genes, which are specifying all sorts of different processes and controlling all sorts of different processes in the embryo. So if mutations occur which change those genes, they're going to change how the embryo forms. They're going to change subtly details within the embryo. And it's those changes in developmental processes which will lead to different outcomes. Yeah. So I like to think of it in this sort of pathway. Differences in genes which control development lead to differences between species. So what do we mean on the left-hand side, changes to the genome? What sort of mutations could be important in changing what genes do in the embryo? I'm going to sort of introduce you what I might call three different ways, three different sorts of mutations. The first one here is a cis-regulatory mutation to a gene. So mutation has occurred in the regulatory elements, as shown by that transition from, if you think of the black box as being the coding sequence of the gene, the little arrow as being where transcription starts from, so the RNA is going to be produced from there. So we have cis-regulatory elements. One of those cis-regulatory elements in this picture has mutated. This is going to change when or where or to what level that gene is expressed. Okay, cis-regulatory change. Another sort of change might be a coding sequence change, a mutation in the part of the gene coding for the protein. This might change. Let's imagine it's a transcription factor. So this might change binding affinity to its site, it might change its actual sequence that it binds to. It might just change something rather subtle like the stability of the protein, what its half-life is, or its interaction partners. Then there's also gene duplication. So mutations can occur where one gene becomes two genes, and then when that happens, there's opportunities both for cis-regulatory change and sequence change after the duplication. And it's this that I'm going to talk about in this seminar, gene duplication and divergence. How important is it to the evolution of development? And can we give some real examples of it playing a role in the formation of different sorts of organisms? So, duplication of genes has been studied by many people. There was a famous book published in 1970 by a Japanese scientist, Susumu Ono, called Evolution by Gene Duplication. And what Ono proposed at length in this book was that extra genes, duplication of genes, gave potential for more complexity. Because if you think about it, one gene is doing not usually one job. One gene is pleiotropic, it may be doing many jobs. But once you've duplicated it, you're sort of freeing up the constraints on that gene a little bit. You may be able to divide functions between the daughter genes. You certainly don't need two genes doing exactly the same job in the cell, so there's an opportunity for evolution to overcome that redundancy and start using a gene in a different way. So how much of this is actually going on? What I've shown in this graph here is the number of protein-coding genes annotated in the genomes of several different vertebrates. So along this phylogenetic tree we go from humans and chimpanzees, mouse, rat, cow, down to zebrafish here, across this tree. This graph, each of these bars, is the number of annotated genes in the genome. Now those numbers, of course, are not going to be superbly accurate. Anyone in this room who's sequenced a genome or tried to annotate that genome will know how much error there's going to be in this. There's genes that exist that we can't see very well, we can't count them very well, and we might count things as genes that are not really genes. But having said that, those numbers are still varying quite a lot. They're varying by thousands between these different, pretty closely related species. So it's pretty inescapable that what we're seeing here is evidence that the number of genes is increasing and decreasing quite a lot through evolution. Okay? The main way that that's going to be happening is through duplication of genes and loss of genes. So we need to think of a dynamic situation where extra genes are being formed and genes are disappearing. So how is that going to happen? Well, there's a number of different ways by which new genes, extra genes, could originate. The first one, that's shown there, is whole genome duplication. In other words, polyploidy effect. So a major error occurs usually in the formation of gametes to form a situation where the offspring is going to have an entire duplication of the genome. Every single gene is double. Okay? I'll talk about that a little bit more in a minute. The picture I've shown there, each of these little boxes is meant to represent a gene. That's meant to be 2 chromosomes. What I just want you to picture from that is that once genome gets duplicated, certain genes will become lost rather quickly, but many will remain 2 copies. So just bear that in mind as we go on to genome duplication. The second one there is tandem gene duplication. So what that means is a mutation where along the chromosome some sort of error occurs, so we end up with 2 genes where there used to be 1. Could be a single gene, it could be many genes, could be a large region, it could even be just part of the gene, but an error has occurred in tandem, in cis to duplicate. Okay, that's a tandem gene duplication. Again, I'm going to give you some examples of that. The third one there is de novo origin of genes. So that's not really gene duplication at all, but it's another way of generating new genes. I'm not going to talk about that. Let's look at genome duplication first of all. So, I first of all want to introduce some slightly older work from my lab, where we were studying this animal here, which I've drawn on this phylogenetic tree, a small animal called Amphioxus, a small marine animal, a few centimeters in length. There are about 30 species of them around the world, and we decided to look at this species, the molecular biology of this species, because of where it fits in evolution. It was already gathering evidence that for certain genes, not very many have been looked at in detail at this stage, Drosophila, fruit fly, might have 1 copy, where vertebrates like ourselves might have more than 1 copy. There was a sort of hint that something interesting was happening here. We thought we could zoom in and sort of analyse what was happening by analysing the genes of this animal, which is an invertebrate branching off close to the vertebrates. So we'll just show you very, very quickly, first of all, the first set of genes we looked at were those Hox genes, the ones that tell cells where they are along the head-to-tail axis. What we were able to show is that whereas in the vertebrates have multiple clusters of these genes are arranged in genomic clusters, many genes in a row, Amphioxus only had a single cluster. And quite clearly, the genes in Amphioxus lined up very precisely to the genes within the vertebrates. So this helped us sort of nail down when the duplication of this chromosomal region happened. But it wasn't just this chromosomal region. I won't show you lots of examples, but virtually every gene we looked at, we were finding one copy in Amphioxus versus multiple within the vertebrates. Here's just one more example. This is a group of genes called parahox genes. There's a cluster of 3 genes in a row. Amphioxus just has a single set, 3 genes in a row. When you move to something like the human genome, then you've got 4 sets. This set of chromosome 13 in humans is from the X chromosome, and these ones are chromosome 4 and 5. Different chromosomes have different sets of these. As we got involved in sequencing of the Amphioxus genome, when we analysed the entire genome, what we were able to show is that across the entire human genome, you could see blocks of 4 copies of the chromosomal regions matching up to single copies within Amphioxus. So what this was evidence for was a growing realisation that at the base of the vertebrates, at this point in evolution here, where I put those 2 circles, the entire genome duplicated and duplicated again. And the outcome of that was that for every gene, it must have gone up to 4 copies, and then some losses will have happened. So some genes are still present in 4, some are in 3, some are in 2, some have gone back to 1. If you look across chromosomes, you can see this arrangement in the human genome very clearly. You can see it in the mouse genome, you can see it across the vertebrates. Work by other people identified another genome duplication here at the base of Helios fish. You see that very clearly in the zebrafish genome and the pufferfish genome. And then there's also evidence now of additional genome duplications happening at various different places in animal evolution. The interesting thing about these is that a genome duplication is really quite a dramatic event. It's going to first of all duplicate every gene in the genome. Even after a lot of loss, you're still going to have a lot of extra genes for evolution to play with and generate new functions potentially. But, as you can see from this rather simplified tree, they're not that common in animal evolution. It's not like they're scattered all over this tree. So if we're really thinking about the influence of gene duplication on evolution, we can't just look at these. I think genome duplications are absolutely fascinating. I'm still working on them. But we really can't just look at genome duplications. We have to think about other ways of generating new genes as well. So I'm going to spend the rest of this talk looking at tandem gene duplications, which are extremely common across genomes. Whenever you start comparing genomes, you see lots of examples of these tandem duplications. So rather than sort of giving you the global view, I thought what would be useful is to just pick 2 examples. I'm going to go through 2 examples in detail about how tandem gene duplication has generated really quite new types of genes which have been recruited for some very interesting functions in the embryonic development of animals. The first example I'm going to show you relates to insects. Of course, insects are extremely abundant organisms, the most diverse phylum, subphylum on the planet. And there are some large groups of insects. The flies, the beetles, the ants, bees and wasps, and the Lepidoptera or butterfly moths are the group of massive numbers of species. The example I'm going to talk about relates to butterflies and moths. So we have to start back with those Hox genes again, the genes which are arranged in a cluster telling cells where they are along the head-tail axis. This is the set of genes in Drosophila. I just want you to look at that for a minute. So here's a picture of a fly, and these 8 key genes in particular telling cells where they are. Now, let's look across insects. Okay. Here we have the same Hox genes from a range of different insects. We've got here from top to bottom a fruit fly, a mosquito, a beetle, a honeybee, and a locust. Okay. You can't read the names of the genes, but you can see the colour coding. What I want you to see is that there's a great deal of similarity between these different species. There's a few minor differences, that on the whole, Hox gene clusters are very well conserved. They don't change all that much in evolution in terms of the total number of genes. Even across the whole animal kingdom, there's a huge amount of changing the size of the cluster. We were therefore really amazed when a paper came out on the Hox cluster of Bombyx mori, the silk moth. And this is a group from China. What they did is studied the Hox cluster of Bombyx mori, and I want you to notice that in between where my red dot is there, that Hox gene, and that Hox gene, an enormous extra number of Homeobox genes, or Hox genes, were found. Why are they doing that? I mean, that's astonishing. Instead of having this in an extremely conserved set of genes, which seems to hardly change across insects, here's a species with a mammoth expansion. Now, all those genes, they're quite different in sequence from the normal Hox genes, but they're still recognizably a divergent type of Hox genes. They clearly come from a tandem gene duplication and then sequence change. They clearly come from right in the middle of it. They can't really come from anything else. So I looked at this, and my first thought is little moths, but they can't even fly. They just walk around. They never survive out in the wild. So there's clearly been human selection on them over millennia for improved silk production, not for being better at being a moth. So I worried, therefore, that maybe what we were looking at was something really peculiar which had happened in the genome of the joint domestication. It wasn't really a natural situation. So an undergraduate student, actually no, it was a high school student visiting my lab, started to do some PCRs on different species of silk moths. And she started to get information that perhaps these genes did actually exist in other wild silk moths. But we could never really get the whole genome sequence at that time. We were just getting clues. So our first real insight into what was going on came when I was asked to be involved in annotating the genome of this butterfly. This is a butterfly called Heliconius melpomene, and it's a South American heliconian butterfly. And a group in Cambridge has sequenced the genome of this butterfly. They were interested in the wing patterning. Okay? I wasn't interested in the wing patterning. I wanted to look at the Hox genes of this. So we annotated, Laura Ferguson in my lab annotated the Hox genes of this species. And here on this diagram, just to orient you, there's the silk moth with all those genes in purple in the middle. And here's Heliconius. We have 4 of these genes. So even in a butterfly, a long way evolutionarily from that silk moth, there's already been duplications and divergence, origination of these genes. So it isn't something really peculiar to domestication. Well, maybe the vast number is, but the origin of the genes is what are known as special Homeobox genes, SHX genes, is not peculiar to silk moths. It's something perhaps found more widely across the Lepidoptera. But how widely? Well, as an evolutionary biologist, you're never satisfied with just 2 species. One reason is that 2 is a pretty small number. The other reason is, if you've got 2 species, you can't actually tell what their common ancestor had. You don't know if there's been gain of genes on one lineage, loss of genes on the other. You need more than 2. So, postdoc in my lab, Ferdinand Marlataz, set about doing low coverage genome sequencing of a range of different butterflies and moths, just so that we could count the number of Hox genes that were there. So we chose a number of different species. We chose 2 other butterfly species. This is the comma butterfly here, Staphyloid butterfly. Then we chose several species of moth. So we've got the scarlet tiger moth over there. This one here, the horse chestnut leaf mine moth, the tiny little moth that I'm going to argue is the commonest moth in the world. If you, across Europe, all our horse chestnut trees, very common tree, are completely infested in this moth. Millions and millions of them on every single tree. So across Europe, vast numbers of this species. And then the orange swift moth, which is a part of the family Hepialidae, a very basal lineage. These lineages of moths haven't even developed many of the features you see in other butterflies. And then as an outgroup, we also sequence the genome of a caddisfly. Caddisflies have aquatic larvae, so they're not butterflies or moths. They're not in the Lepidoptera, they're in the Trichoptera, but they're the sister group to the Lepidoptera. So what did we find in relation to these genes? So just to orient where you are, on this diagram, we have the Hox cluster at the top up there, and we're zooming in to a little part of it. The bit we're zooming into are these three genes called Proboscipedia, Zen, and Deform. Okay? So here's the heliconias that you see before, Proboscipedia, then there's SHX and special Homeobox genes, then we go Zen, Deform. And here's Bombyx, with lots of these SHX genes. So what do we find in different species? First of all, the caddisfly. The caddisfly was completely normal, no trace of these genes at all. Then we move to the orange swift moth, which is basal lineage. What we can see there is it's pretty normal. There's something strange is going on with this gene, but pretty normal. But no trace at the sequence level of those SHX genes. But as soon as we get to the rest, we almost always find 4 genes, just like in the heliconia. So the heliconian butterfly was a pretty good example of what most of the butterflies and moths look like. Bombyx are still gone weird. But essentially, what we're saying is at this point, rather early in Lepidoptera evolution, but not right at the base, not right at the start of the butterfly evolution, pretty early, one of these Hox genes must have tandemly duplicated to generate new genes, which then changed its sequence quite a lot. So what do I mean by changing sequence? This is an evolutionary tree. Take a bit of staring at to see what you're looking at here. This is an evolutionary tree of amino acid sequences from different Hox genes across the butterflies and moths. First thing I want you to look at is down here. If you look at a gene like ABDB, which is one of the Hox genes, the thing I want you to notice is that those little branchlets there, those tiny little dots, are the rate of amino acid chain. They're hardly changing at all. And it's the same all the way around these traditional Hox genes. Very, very conserved sequences, fairly changed between the Lepidoptera. But as soon as you get to SHX genes, long branches. And this tells us that after the origination of these genes, evolution has been very rapid, changing their amino acid sequences, and seems to have continued changing their amino acid sequences even after the butterflies and moths like those. They're fast evolving genes. The other thing you might be able to see from this is that the closest of the Hox genes to the SHX genes is a gene called Zen, which was one of those sitting right next to them. There's Zen in yellow. So that makes perfect sense that they seem to have come from Zen and duplicated the population sequence. So what are they doing? Well, the first thing that we were slightly worried about was you look at those branch lengths, and they're evolving so quickly, I wonder whether they would still be able to function as a homeodomain transcription factor. Because what I haven't told you is the Hox genes are regulatory genes. They have a particular fold in them, a particular protein fold, a helix, loop, helix, turn, helix fold, which allows the protein to dock in a double helix of DNA to switch genes on and off. And that's a pretty conserved fold. So if these genes have changed rather dramatically, could they still fold? Well, we haven't done a biochemical test of that, or a crystallography test of that, but we have done molecular modelling of it in collaboration with Willie Taylor at the Crick Institute in London. What you're looking at here is a three-dimensional model built from the amino acid sequences of one of these homeodomains. So this is the DNA structure that it should lock into. There's the three-dimensional fold. So the first thing we're able to show is that for every one of those SHX genes, molecular modelling says it's perfectly capable of folding into 3 alpha helices of the right size and shape to dock this DNA. Okay, so I won't show you every structure, but the structure prediction says they're perfectly capable of doing that. But would they bind DNA in a sequence-specific way, which is what we need for a transcription factor to actually activate or repress genes? Again, what we did for this was something that Willie sort of invented. I was quite surprised how well it worked. We tried to do it in silico evolution experiment, evolution in the computer. So what we did is we built structures where the DNA to which the protein is binding starts off as a complete homopolymer run. So A. On the other strand, of course, will be T. And we bind in the computer the predicted protein to that DNA molecule. And we calculate binding energy.
Then we start mutating the DNA sequence, not the protein sequence, the DNA sequence that it's binding to, keep mutating it, and we select the best binding profile. And we mutate again, and we select the best binding profile, etc., etc., etc. We do that lots of times. And what comes out of that is evidence. It should, if it has sequence-specific DNA binding protection, should converge towards a preferred binding sequence. And that's what we're able to do for all these. These are the SHX genes from the speckled wood butterfly in A, B, C, D, E. And each of them converge to a subtly different binding structure, binding sequence of DNA. Now, that doesn't necessarily mean that that protein will necessarily bind to that particular DNA sequence, because we're doing this in silico, we haven't got partner proteins there, we haven't even got that entire homeodomain protein, we've just got the one part of it which forms the homeodomain. So we're sort of slightly out of context of the real cell, but I think it is giving us rather strong evidence that these proteins will most likely bind DNA in a sequence-specific way. So we're really encouraged by that. But what do they actually do? Well, functional tests are very, very hard to do left on at the moment. They're only just starting now. So our only clue really came from gene expression. And the gene expression was amazing in these genes. The gene expression patterns we saw were just mind-blowing. What I want to show you is this. There's a lot of information on this slide. We're looking at SHX A, B, C, D. E is that Z gene. Start at the bottom down here, and I'll show you what all this means. What you're looking at the bottom of developing embryos of butterflies. This was work done in collaboration, I should say, with Casper Brucker at Brooks University in Oxford. What you're looking at there, you're seeing staining primarily in a layer of cells which envelops the embryo. That layer of cells is called the serosa. It's an extra embryonic membrane, an extra layer of cells. So it has some very important functions in the development of insects. To do with protection against desiccation, immune defense, all sorts of things. So you can see the genes expressed there. As we go earlier in development, now look at the middle row there. What you're seeing there, that strange shape, is the development of that serosa, very specifically, because the cells divide and then grow around the embryo. So you can see that happening. It starts as that strange sort of egg-timer shape and then grows out. So you see that very beautifully. If you go even earlier, there's the most amazing one, which is this one here. What you're looking at there, and I will zoom into it, is the unfertilized egg of a butterfly, still inside the mother butterfly. So this is the ovaries, the ovariole, and each of these is developing an egg. So on the left-hand side there are cells, which are called nurse cells, which are provisioning the egg. If you follow my red line, that delineates one cell, and that's the unfertilized egg. So even within the unfertilized egg of the butterfly, the RNA is localized in this amazingly complex pattern, which is marking the part of the egg which, after fertilization and after nuclear division, those nuclei will eventually, after cellularization, form the serosa of the embryo. So it's prefiguring where the serosa will form. So it seems, even from the expression pattern, pretty clear that these genes are going to have a function in specification of that serosa. And I think this is also probably one of the most complex RNA localizations that's ever been described within a single cell. So, what does that mean for being a butterfly or a moth? What does it mean what does it mean from an evolution perspective? Because what we got here is early on in the evolution of butterflies and moths, a Hox gene duplicated, tandemly duplicated, changed quite a lot in sequence, and has required a new job. That new job seems to involve in specification of serosa. So how do we put all that together? Well, I'd like to present a hypothesis about what I think is going on here, which is, I did mention that the serosa has these very important jobs in desiccation defense, immune defense, etc. Well, during the evolution of butterflies and moths, there have been some very interesting ecological transitions. One of the most dramatic has been a transition for butterflies and moths to lay their eggs on leaves. So if you were to watch, let's say, a swallowtail butterfly today laying eggs, they'll be laying eggs on leaves, exposed to the environment, exposed exposed to the wind, exposed to desiccation. But the basal species don't really do that. So remember that swift moth, the Hepialid? They don't do that. Hepialids lay their eggs in soil, wet soil. They don't have the desiccation problem. And then the larvae, when they hatch, eat roots of developing plants. If we go to the outgroups of the Lepidoptera, the Trichoptera, the caddisflies like that, remember they didn't have these genes. Their eggs and larvae are aquatic, completely living in rivers and streams, so they don't have that problem as well. So I would hypothesize that what's happened here during the evolution of Lepidoptera is the evolution of new genes which were used for a new job, which was to somehow specify new functions of that serosa. Okay, maybe enable it to develop better desiccation resistance, potentially better resistance against parasites and pathogens as well. So we may have a link between genome evolution and phenotype evolution there. Okay, let me move on to the second example that I want to go through, which is not insects now, it's mammals. So we're getting a bit closer to home. This is work done primarily by two postdocs in my lab, Nacho Mizo and Thomas Dunwell, but I'll show you other people involved in this as well. We start with those Hox genes again. And remember I said the Hox genes code for transcription factors, so the protein sequences have this particular fold. Well, actually, Hox genes are not the only genes in the genome which encode for proteins with that fold, with that homodomain fold. There are other genes in the genome, and there's a lot of them. So, in the human genome, there are more than 200 genes which encode for proteins with that fold. So we call that collective group of 200 genes the Homeobox genes. And within the Homeobox genes, the Hox genes are just a small part. They're just 39 of them. Okay, so those are the sort of numbers. So this, what you're looking at here, you can't see it very well, is a phylogenetic tree of all the homeodomains in the human genome. So when the draft human genome was first published in 2001, I had a new graduate student in my lab, Anne Booth, and I asked her, I was thinking, what can I ask a new graduate student to do, which won't take too long. So I said, why don't you just make a list of all the Homeobox genes, there's the human genome sequence, make a list of all the genes and all the Homeobox genes in the human genome. I thought it would take about 3 months. It took 3 years. The reason for that is I was actually really surprised that there were a lot in there weren't known. I thought they would all be already known. Because when, for most gene families, we knew a lot of genes before the genome was sequenced. We just didn't know quite where they all were, which chromosome, exactly how they were arranged. But it wasn't the case here. So we were able gradually, over a number of years, to put a list of all the human Homeobox genes together. Now, most of them were well known, but there were a few which were not. A few were completely novel. They'd never been seen before, they'd never been sequenced before, they didn't even have a name or anything. Okay? Let me just mention a few of the key ones that Anne found. The first one was this one. The nice thing about finding a new gene is you're allowed to give it a name. That's always, it's usually just taxonomists, like species, that get named things. Molecular biologists don't. This was exciting. So we have a gene here. What I'm showing you is just the amino acid sequence of the homeodomain part. Okay? Not the whole protein, just the homeodomain part. When I looked at the homeodomain sequence, it seemed rather strange because in this part here, that part where I'm sort of waving the red dot, is the bit which is helix 3, which is the bit that locks right into the major group of DNA, the most critical binding protein. And right at the centre of that, there was a critical amino acid change to an arginine. So single amino acid might not sound a lot, but that position 50, is very important. So we call this arginine 50 Homeobox. Here's the next one, which we call leucine 20 Homeobox. You might see there's a pattern emerging here. So this, again, is a rather critical, usually well-conserved site. This is actually in helix 1 now, where there's been a very unusual amino acid change. The next two genes, the homeodomain was rather divergent, but there was something peculiar outside the homeodomain, which was 4 amino acids which seemed to be repeated again and again and again
call these tetrapeptide repeat homeobox genes, of which there were 2 in the human genome. And then there was another strange and divergent gene, but we'd run out of interesting names, so we called it divergent paired related homeobox. So we had these new genes, very exciting, gave them names. But remember, at this stage, we didn't know what they did. They're just there, sitting in the human genome. We also didn't really know where they came from. They clearly come from another homeobox gene. They've got a homeobox motif in. Clearly come from another homeobox gene. They're very, very divergent in sequence, rather different in sequence from all the others. Okay? But we had a clue. One clue was chromosomal position. Here's these genes, RGFX, DPRX, TPRX1 and 2, and WHX. There is another gene here sitting near to them. That gene's called CRX. CRX stands for cone and rod homeobox. It's a gene involved in development of the retina. And that was a very, very well-known gene, very well characterized across the vertebrates, involved in eye development in all the vertebrates. It's a homeobox gene. It's quite different in sequence from the ones that I'm talking about, but it is a homeobox gene. So we had a hypothesis that maybe CRX had tandemly duplicated and given these genes. Now, TPRX1 and 2 are pretty close to CRX, so maybe that's not too surprising. But the other ones are a very long way away down that chromosome, chromosome 19, or even RGFX is on chromosome 3, a different chromosome altogether. So we needed a little bit of imaginative thinking to come up with that hypothesis. Didn't really have very much proof. But much, much later, we were able to prove this. And this is work of Nacho Maito, who joined my lab as a postdoc many, many years after this. The reason that he was then able to prove that that hypothesis is correct was because in the intervening period, the genome sequences of many, many mammals have been published. We didn't just have the human genome and the mouse genome to work with, there's lots of genome sequences. And using phylogenetic trees involving lots of mammals, but also positions of conserved non-coding elements, which are also quite good indicators of relatedness between genes. I don't want to go through that data in detail. We were able to show this, which is that CRX has definitely undergone tandem duplication and given rise to these genes. Okay, so it's undergone tandem duplication, and then the sequences have changed. Okay. When did it happen? Well, again, remember, there's all those sequences now of genome sequences from lots of different animals. This analysis of these, again by Nacho, showed very clearly when they happened. All this happened at the base of the placental mammals. If you go outside the mammals, you don't find these genes at all. You find the CRX gene, but all the other ones are not there outside the mammals. And in fact, even if you step inside the mammals, the platypus doesn't have them, the molluscs don't have them, the echidna doesn't have them. The marsupials like kangaroos, wallabies don't have them. It's just the placental mammals which have them. So at the base of placental mammals, we can say that the CRX gene underwent those duplications, gave rise to these genes. Okay? And you can see that by these are a range of different species that we've looked at here. Now the other thing that you might notice from this is that the organisation of these genes is pretty different between different species. They don't all look like the human situation, the human ones at the top. Some of them have got extra genes, some of them have got genes missing. There's lots of extra duplication happening. So these blue arrows point out that that TPRX2 gene there has duplicated 67 times in the mouse genome. Not, some of them are now super genes, going crazy. This one down here 6 times in 10 rep, that one 14 times in a guinea pig. These genes, this looks like, these genes, they didn't just, we shouldn't just think of it as a gene duplicating tandem. We should think of this as a much more dynamic situation. That at the base of mammals there were duplications, and they've continued to duplicate. And in parallel with the duplications, there were losses as well. So all those red crosses are where we know genes used to be, but they've gone. Okay? We can infer they used to be there by comparison of genomes. So even the human genome lost one over there. Okay? So there's been lots of gain and loss of genes, all happening through mammalian evolution. Really, really dynamic. So I was very surprised by this because most of the regions of the genome, I've done a lot of staring at genomes, and you don't normally see this. You look across a group of pretty closely related species like mammals, you pretty much see similar organisation of genes. So I never see a genomic region like this. This is primarily on chromosome 19 of humans. near these genes, there are plenty of other non-homobox genes, things to do with all sorts of other things like immune function and all sorts. And the more we look, we realise they're doing this as well. This whole region is chromosome 19. You see, it's very, very, very important. So I went to see a colleague of mine, Professor Gil McVean. He's at the University of Oxford. He's a real expert on the human genome. He's expert on recombination and variation between species. So I asked him, well, isn't this a little bit strange? There seems to be something, what you might call a hot spot on chromosome 19. Something funny is happening here. And he said, yes, 19, the devil's chromosome. So what do you mean by that? What he really meant was that to human geneticists like him, chromosome 19 always created problems. One which was really hard to sequence, it was really hard to assemble, it was really hard to study in all sorts of different ways. Let me give you two published bits of information from other groups which back up what I'm just saying. On the left-hand side, here, this graph shows human chromosomes, 1, 2, 3, 4, etc., down to the X and the Y chromosomes, and the density of genes per megabase. You'll see a great big spike there. That spike is chromosome 19. Genes are packed into the chromosome. That is definitely the result of tandem duplications, because a lot of that spike is due to arrays of genes. So there's clearly something happening here. The graph on the right is a little bit harder to understand. What you're looking at here is the distribution of recombination sites during meiosis on different chromosomes. And without pulling that graph apart too carefully, I just want to point out that the more each one of the lines is a chromosome, the more the graph is squished to that side, it's the more that the chromosome relies on recombination hotspots. There are particular sites on chromosomes which preferentially undergo crossover. Okay? There's one line that is an outlier from all the rest. That one there. No prizes for guessing which chromosome that is. That's 19. So it looks like recombinations and meiosis will distribute in a lot less hotspots. I suspect that gives it more chance to go wrong and maybe make incorrect crossovers, which actually will cause tandem duplicated genes and deletions of genes. So it seems to be all fitting together there mechanistically. But I still haven't told you what they do. That's because it took a very, very long time, more than 10 years, to get any clue as to what these genes really do. So again, let's just see where we are. We've got a gene, CRX, which as I said, is primarily involved in development of retina, is duplicated to give these different sorts of genes in the placental mammals. Well, right at the start, even when Andrew, the graduate student, was doing this work more than 10 years ago, we had one clue as to what these genes might be doing, but it was a very, very small clue. The clue comes from something called pseudogenes. Okay, a pseudogene is a dead copy of a gene, a non-functional copy of the gene. There are two different ways in which a pseudogene can form. One is called a duplication pseudogene. A duplication pseudogene is some sort of duplication event happens, and then mutations happen in that gene, and it stops being functional. So forget about those. They're not relevant to this one. What's relevant to this one is the other type of pseudogene. This is called a processed pseudogene. A processed pseudogene is a gene that's transcribed into RNA, and then reverse transcription enzyme, which is kicking around our cells, accidentally copies that RNA back into DNA and puts it in a chromosome somewhere. It's usually a dead copy of the cell because it hasn't got a promoter, hasn't got enhancers, it's just been put randomly into the sequence and it accumulates mutations and dies essentially. Now, those genes that I described to you, those new mammalian genes, tended to have a lot of pseudogenes. We had spotted that processed pseudogenes. So that gave us a clue as to what they did. Why am I saying possession of these has anything to do with function? We have one link, which is as follows. Think about gene expression. Imagine a cell on your shoulder, let's say, expressing a skin cell. It's expressing a gene there, expressing a gene, RNA. The reverse transcriptase, if there is any in that cell, might copy it into DNA and put it in some on a chromosome randomly. Okay? That copy, although something's gone wrong in your cell, is not going to be inherited to the next generation. Because you don't make children from your shoulder. So, it's completely irrelevant to genome sequences. It's completely irrelevant to evolution of genome sequences. So we had seen copies of these genes in the human genome. If, however, the gene is transcribed either in germline, testis or ovary, or indeed in very early embryonic cells, if the transcription happens there, copied into DNA, integrated back into the genome, that will be in the next generation and will ultimately contribute to the human genome if your descendants conquer the world. So, because our genes that I'm describing to you have these processed pseudogenes, they must, by definition, be expressed somewhere in the germline. You knew that. So we started to look. We searched every germline tissue we could find. We got human embryonic stem cells, no expression. We got testes from the hospital, no expression. So we were starting to question our logic. We really couldn't find anywhere where these genes were expressed. Here's a whole range of human tissues, so embryonic stem cells, a whole range of tissues. And here's our genes we're interested in. Blue means the gene is not expressed. There's quite a lot of blue on that slide. Occasionally there's something which isn't quite so dark blue. There might be one or two transcripts there. It just didn't look like anything. We're a bit about to give up at this point when a number of groups published some very interesting transcriptome data from the pre-implantation human embryo. And that's where we found these genes expressed. So the genes are expressed in the pre-implantation embryo, coming on mostly at the 8 cell stage, staying on to the 16 cell stage, and then going off. And that's it for the rest of life. Okay? So a really sharp pulse of expression. It's not just human. Remember, these are mammalian genes across all the placental mammals. There's data, expression data of cow homologs of these genes. One of the genes decorates as a pseudogene in cow, that doesn't seem to be expressed. The others are expressed at 8 cells or 16 cells. So we've got a clue here from expression that whatever they're doing it then, pre-implantation, where the cells, before cells have even formed the blastocyst, totipotent cells. So studying the functional biology of pre-implantation cells is nearly impossible. That's the problem. These are almost inaccessible cell types in any species. So we had to do a bit of a proxy. So just in the last couple of slides, I want to show you this one experiment. What we did, and this is Thomas Dunwell, postdoc in my lab, we made V5 tagged constructs of each of the human genes, and we ectopically expressed them in adult cells. We took primary adult cells, and we asked the question, if we switch one of these genes on in an adult cell, which should never be expressed, are we going to influence the transcriptome of that cell? And if we influence the transcriptome, can we make any sense in it? Is there any sense of the changes that happen? Anything embryonic sort of happening? So we did this, did RNA-seq. Lots of genes go up, lots of genes go down. I don't want to go through all these numbers, but there was a very interesting pattern in there. The pattern that we could see was that 2 of the genes, LUTEX and TPRX1, had quite a large overlap in the genes that they regulate. The others didn't really. Quite a lot of overlap between those 2. Down-regulating the same, pretty much an overlapping set of genes. And interestingly, one of the other genes, that RGX, gene 50 had an overlap in its upregulated genes. So over 100 genes here, which are upregulated by RGX, downregulated by the other 2. None of the other combinations that you look at have anything looking like this. Something really jumped out at us. But what are those genes? Well, the question you really want to ask is that do those genes have anything in common with genes expressed in the early embryo? So an interesting analysis was suggested to us by a collaborator in Barcelona, Manuel Irimia. What Manuel had been doing is studying gene expression in the early human embryo. Taking all the data sets published for gene expression, 1 cell, 2 cell, 4 cell, 8 cell, etc., all the genes in the human genome, and categorize them into what we might call temporal expression profiles. So that means genes that go up, stay up, genes that go down, stay down, genes that go up and down, up, patterns. He found about 100 different patterns, as shown here, of different patterns. And then we asked, any of those patterns overlap in genes. Each of these patterns, of course, is a set of genes. Any of these sets of genes overlap with the genes that we're regulating when we ectopically express our genes. And the answer is yes, one. Just one. This one jumps straight out with a very strong, statistically significant overlap with the LUTEX down, TPRX down, and RGF up at centre of that Venn diagram. And what that pattern is, are a whole set of genes which in a human embryo go up at 4 cells, so they're start in between 4 and 8 cells, and then up at 8, they're up at 16 for borreliola, and then down that last 6. So it's mimicking the expression pattern of genes themselves, but it really does look like what we've got there is a set of new Homeobox genes which have originated by tandem duplication and got a new job of activating a spike of expression of lots of genes in the human early embryo. We don't quite know what that's doing, but it's clearly something quite striking in the development of mammals. And we note that this is absolutely the time just before cell decisions are made. The first cell decision that has to be made in the embryo. Remember I said right at the start, big problem in development is 1 cell, 2 cell, 4 cell, 8 cell, etc. The cells have to decide what they're going to become. The very first decision they have to make is between embryo cells and trophectinum cells or placenta cells. The very first decision they have to make. And it's just before that decision happens, this pulse of expression happens. So our working hypothesis is that these new genes originated and they've been recruited for regulatory purposes just before that critical cell decision, maybe preparing cells for that critical cell decision between embryo and placenta, which of course is actually a really key feature of being a placental mammal anyway. So perhaps it's no wonder these is correlating with the origin of the placental mammals. So, to conclude, what I'm trying to show you today is that for a long time we've seen an extreme amount of conservation of genes between different species, revealing deep similarities in the development of different animals. But those genetic systems do change in evolution through mutation. The sorts of changes we see are a great variety. The ones I've talked about are whole genome duplication, which we see in vertebrate evolution, and tandem or single gene duplication, and I've given you examples from insects and mammals, just as worked examples we've looked at in detail. Many other examples are out there. And I think we're sort of just starting to scratch at the surface between what I call the genotype-phenotype interface, evolutionary changes in the genes, and how that links to evolutionary changes in the whole organism, such as the egg of the butterfly or the embryo of a mammal. And with that, I want to thank you for your attention, and I'll stop there. Thank you.
I'm sure Professor Holland happy to answer a couple of questions from the audience. Maybe I ask the first question. It's very interesting that in a particular, Homeobox is activated in 4 cells and 8 cells. But because I read somewhere that the 4 cells are actually not exactly identical. So probably be experimentally difficult to see whether which of the 4 cells they actually express.
Yeah, we don't. Yes, you're quite right. Transcriptome sequencing of individual cells, which is being done, single cell sequencing, is revealing differences. We don't really know how stable those differences are, or whether sometimes there are temporal fluctuations in gene expression. So it's a very difficult thing to address. Humans are not going to be the right species to do that on. Mice are also not going to be, because of that crazy duplication. One of them has duplicated 67 times, and other ones have been lost. So we have to find a different model to work with from amongst the mammals. But it could be that they're preferentially in particular cells. We don't have any evidence of that.
Mammals here, but I'd like to ask a chicken and egg question. The chicken and egg question is, these are transcriptional factors, the Hox genes. So how about the targets? Would there be the new targets appearing that demands the new transcriptional factors, or vice versa?
We have tried to look into that because what we could do, of course, is we have an estimate of what some of the targets may be, because of genes that respond transcriptionally. We've tried to see if there's an excess of new genes in there. We haven't detected an excess. I'm not saying there aren't some new ones. We haven't detected a dramatic excess. So I think that new genes and the divergence of a new transcription factor will have some effect by modifying the expression of existing genes to start with, because you can imagine situations where even subtly affecting the expression of pre-existing genes could have quite a phenotypic effect. And then with a new transcription later on, perhaps new targets come in. So I think, in answer to the chicken and egg question, I think transcription factors first. Okay. Yes, go ahead.
Thank you, Professor. I wonder if any of these, the triple X, those few genes control the epigenetic programming, because they are expressed as the in cell or tubular stage.
Yeah, I think, so the question is whether some of the targets of those new mammalian genes affect epigenetic characteristics of the early embryo. I think the answer is yes. We haven't proven that definitely, which we would have to do by actually profiling epigenetic marks, histone modification marks, for example. But amongst that series of genes which go up and down are some histone modifiers. Okay, amongst the genes, and many other genes are changing, metabolic genes, etc. But amongst them are some histone modifiers. So I think you're right. I think some epigenetic changes will be being driven by these genes, but we haven't actually proven it yet.
Yeah. Any more questions? Okay, one at the back.
Organization, could you demonstrate a point that why you have that kind of pattern? I mean, you predict that it may be related to protection of the eggs, but why you have that kind of special pattern?
Well, I think spatial patterning, localization of RNAs, is quite a common mechanism in insects to get very rapid formation of particular tissue types. Localize the SHX RNA in that way very early is to ensure that the serosa cells can be specified very, very early and start developing. That's what I think the reason is. Doesn't answer the question how it gets localized like that. We really don't have a clue to that. But as an answer to your question of why, I think it's to do with speed of development.
Yeah, rather technical question. So when we look at these duplicated genes, the Hox genes, the new ones, so would we consider them as old genes when we look at the phytostratum kind of classification? So could we consider them as old genes because they exist in many organisms along the phylogeny, or should we consider them as new genes?
Okay, so what is a new gene? Is essentially the question. Some people will be very strict and say a new gene has to be a de novo gene that comes from non-coding DNA. I personally think that's too strict, because that's not how biology works. I think a new gene is something doing a new function, really. So my idea is if there's an old gene which duplicates and one of the copies becomes radically different, I would prefer to think of that as a new gene. Even though its parental gene is there, if it's changed a lot and there's evidence it's doing something new, I think it's just more helpful function to think of it as a new gene.
Okay. Okay, one more. Yeah.
Thank you. I just wonder, like, for they're both genes duplication, but for some butterfly, they seem similarly they look alike. But for human and mouse, they seem really different in their appearance, I think. So how the genes duplication or some new genes are related to their characters of their appearance?
Right. Well, I think there's two parts to that question. The first is, it might be that those butterflies look alike to us because we're mammals. And the butterfly might be looking at us and thinking, those humans and mice look really similar. part of it might be perspective. Okay, we have to be careful of that perspective. The other is, of course, that what I'm describing here is just a little snapshot of just a few genes in the genome. And we have many, many thousands of genes there. So other genes will have been changing as well.
Okay, any more questions?